Hi, everyone. Thank you for joining our webinar today. We have a lot to cover, so let's get started. We're going to break this webinar into two sections. During the first portion, we're going to talk about what DRAGEN is, what it does, and how to set up a run for analysis. This includes discussion about what is DRAGEN, the common secondary analysis output files, our run setup options. We are going to briefly discuss DRAGEN on the NextSeq 1000-2000 versus DRAGEN on a standalone server. We'll talk about V2 sample sheets. We'll talk about kicking off an analysis by requeuing. And we'll also talk about using custom genomes. The second portion of the webinar will focus on what to expect once DRAGEN analysis completes. This section includes discussion on the following topics, which is where to find your DRAGEN output files. We are going to talk about the NextSeq 1000-2000 analysis pipelines and go through some details there as well. And then finally, we'll end the webinar discussing what DRAGEN output files are going to be transferred by Universal Copy Service, which is from the instrument to wherever our final locations are going to be. So first, DRAGEN stands for Dynamic Read Analysis for Genomics. And of course, our focus is DRAGEN on the NextSeq 1000-2000. Which from here on out, I'm just going to refer to it as the NextSeq 2000. DRAGEN produces common analysis files like FASTQ, SAM or CRAM, gVCF files, and alignment and variant calling metrics. DRAGEN on the NextSeq 2000 also automates analysis. This means that analysis automatically launches on instrument or in the cloud after the sequencing run completes. So what exactly is DRAGEN? DRAGEN is software. DRAGEN contains several pipelines on the NextSeq 2000, including the DRAGEN-GATK Germline pipeline. The DRAGEN Germline pipeline was renamed to DRAGEN-GATK Germline pipeline. This pipeline does not use the GATK variant caller. It uses the DRAGEN Germline variant caller, which is also going to be the case in the DRAGEN Enrichment pipeline. The name change is mainly to reflect Illumina's collaboration with the Broad Institute, which are the developers of the GATK variant caller. We also have the DRAGEN Enrichment pipeline, which is very similar to the DRAGEN-GATK Germline pipeline, except now our focus is going to be in a targeted region. And for RNASeq experiments, we have the DRAGEN RNA pipeline, which performs alignment, quantification, and fusion calling. Lastly, and not listed, we do have the FASTQ generation pipeline. This pipeline uses BCL Convert to generate FASTQ files and corresponding metrics. DRAGEN is also hardware. DRAGEN uses field programmable gate arrays, or FPGA cards. These FPGA cards are also known as the DRAGEN chip or the DRAGEN card. The DRAGEN chip is programmed to execute computing tasks like alignment, variant calling, and counting for the DRAGEN RNA pipeline. Together, the optimized DRAGEN software pipelines and the DRAGEN chip make up DRAGEN. So why is DRAGEN so important? The answer is that it's super fast and it's super accurate. Using the DRAGEN software and hardware accelerates secondary analysis tasks. DRAGEN is up to 6 times faster than Local Run Manager, which is our other on-instrument analysis software found on our benchtop sequencers. Accuracy is not sacrificed for speed. In these charts, we see that DRAGEN had fewer incorrect and missing variant calls both for SNV and endil detection. The accuracy information comes from a benchmarking comparison, so variant calls were compared to an NIST truth set to identify false positives and false negatives. BWA-GATK was the previous industry standard alignment and variant calling pipeline developed by Broad. The DRAGEN algorithm improvements between V2 and V3 increased variant calling accuracy, both for SNV and indel detection, by reducing false positives and false negative calls. So here, when we talk about DRAGEN V2 and V3, we're referring to the DRAGEN software versions, not the DRAGEN server hardware versions. So DRAGEN V3, the software, performs better, meaning there is a reduction in false positives and false negatives, both in SNVs and indel, when compared to the BWA and DRAGEN V2 pipelines. So on the NextSeq 2000, we are using DRAGEN V3. Okay, so now that we know what DRAGEN is, let's talk about the files and the files that it outputs. DRAGEN generates the same types of files as most other alignment and variant calling pipelines, both Illumina and third-party pipelines. So these file types are bands and VCF, so let's talk about these a little bit further. So DRAGEN outputs alignment results in BAM or CRAM format. These output formats are common and are output by most aligners, both Illumina aligners and other third-party aligners. BAM and CRAM files contain alignment results. BAM stands for Binary Alignment Map File. A CRAM file is a compressed BAM file. BAM and CRAM files are in binary format. So the example that we have here of a BAM file converted to text format is not really typical. We need to use a tool in our Linux environment like SAMtools to get this text output. Most people will not visualize a BAM CRAM in this way. So instead, they'll load the BAM and the CRAM into a genome viewer, such as the Broad's Integrative Genomics Viewer, or IGV. Each blue box marks an entry in the BAM or CRAM file. Each entry contains information about an aligned read, including the read name, which comes from the FASTQ file, where the read aligned, where the read's mate aligned, so the equal sign here means that the read and its mate align to the same chromosome, the read sequence, and also the read's base call quality score. And here it's encoded with ASCII characters, and each character represents a numerical quality score. The Broad's Integrative Genomics Viewer, or IGV, can be used to visualize alignment results in BAM or CRAM files. So the screenshot here shows a BAM file from an enrichment sample loaded into IGV. The gray bars represent aligned reads. The colored ticks within the gray bars represent mismatches between the aligned reads and the reference sequence. These might be real variants in the data, but we would actually have to go through variant calling to verify that. The reads shown here align to the EGFR gene. At the bottom, the blue boxes represent exons, and the blue lines represent introns of the EGFR gene. So the arrows show the direction of transcription. Along the top of the screenshot is a coverage histogram, again in gray, which shows the higher coverage of exons, which we would expect in enrichment data since we target those regions. Okay, so let's talk about variant calling results. So variant calling results are written to standard variant call format, or VCF files. DRAGEN uses VCF format version 4.2. A VCF file contains information about all variant positions identified in the genome. A VCF file is a list of variants identified. So each line or record in the VCF contains information about an identified variant. A genome VCF, or a gVCF, is larger than a VCF and contains information about all the variant positions identified in the genome, just like the VCF, along with information about positions identified as reference or not variant in the genome. This information can be very useful, especially if you're expecting a certain variant but it wasn't called. You can definitely check a gVCF file to see what's going on in that position. So here in this example, we have an example VCF file. So let's talk about these columns. The columns highlighted in the purple are the chromosome and position. So this is going to indicate the exact location of the variant. The columns highlighted in teal are going to be the ref and the alt. So the ref represents the base call at the reference genome, whereas the alt column is going to represent the base call at that position in the sample. The green column represents the filter column. In this case, all of these variants here are passed, but there are instances when variants may not pass. If that's the case, in the filter column, we'll indicate which filter was triggered that stops that variant from passing. The next two columns, the format and the sample column, they work together to give us information about our variant. So the first column highlighted, or the first section of the column highlighted here in yellow, is the genotype. So we see the GT in the format and then the actual call in the sample column. So for the first one, the genotype, for the first variant listed here, the genotype is 1,1, meaning that it is a homozygous variant call. The next section, the AD or allelic depth, gives us the depth of both the ref, so the reads supporting the reference call, and the reads supporting the alt call, or the variant call. So again, looking at the first example, we have a 0 and a 9, meaning that we had 0 reads supporting reference and 9 reads supporting the variant. Next in red, we have the AF, or the alternate allele frequency. So again, looking at that first example, since all of those 9 reads are supporting the variant call, we're going to get a variant allele frequency of 1, or 100%. And then lastly, this gray column here represents the depth. So this is going to give us the total reads, regardless of which they support, that align to the specific position. Okay, so now we have an example genome VCF or a gVCF file. A gVCF file, again, lists information about variant and non-variant positions in the sample. So it contains more information than a VCF. In this example, there is actually one variant reported at position 6279370. I'm just going to highlight it here for you to see. Samples will generally contain large stretches of non-variant sequences. So rather than report each non-variant position individually, stretches of non-variant sequences with similar read depth and are reported as blocks or bands in the gVCF. The blocks of non-variant sequences start at a position listed in the position column, or the POS column, and end at the position listed in the info column. In this example that I have highlighted, we have a block that's 25 bases. Now, non-ref is a symbolic allele listed in every record's alt field. This represents the possibility of having a non-reference variant allele at each site. So again, just like we saw with the VCF file, the gVCF file also has the exact same columns, position, ref, alt, filter, and the format columns. But what we want to keep in mind with the gVCF file is that if we have at every position that we did not identify a variant, but it's actually ref, that's also going to be listed here, and we can interrogate that further if we need to troubleshoot. Okay, so now that we know the file types that DRAGEN produces, let's talk about the run setup options for the NextSeq 2000. Okay, so we have 4 different run setup modes. First, we have standalone mode. So for standalone, no DRAGEN analysis occurs on the instrument or in the cloud. Next, we have local and hybrid modes. In both cases, DRAGEN analysis occurs on the instrument. These two differ in where the run is set up. So either you're going to use a sample sheet for local only analysis, or we're going to set up the run in BaseSpace for hybrid mode. Regardless of where the setup is, the analysis still occurs on the instrument. Lastly, we have cloud-only mode. So for cloud mode, DRAGEN analysis occurs in the cloud. The run setup is also done in BaseSpace. Okay, so now that we have an idea of the different run modes that we had, we just briefly want to talk about the difference between DRAGEN on the NextSeq 2000 versus the standalone server version of DRAGEN. Okay, so we talked about the different analysis modes, but I want to mention what exactly happens with each of these options. So when analysis is occurring locally on the instrument, on board the NextSeq 2000, the multiplexing starts after index reads complete using DRAGEN. Preliminary alignment of read 1 sample data to the reference genome starts right after demultiplexing complete, again, using the DRAGEN on the instrument. In cloud analysis, demultiplexing and secondary analysis do not start until the run completes and all primary data has been uploaded to the cloud. Also note, cloud mode does not mean secondary analysis is performed by BaseSpace apps. Okay, so now if you're using a standalone DRAGEN server for analysis, This means that it's not going to be on the instrument, and you're actually going to transfer the data from the instrument to that final server. Now, in using a standalone server or a standalone DRAGEN server, demultiplexing and secondary analysis can be started manually after the run completes and all primary data has been transferred over to the server. All right, so now that we are aware of the different run modes, how analysis starts locally on the cloud and using a standalone server, let's talk about generating V2 sample sheets for local analysis. So we recommend using the instrument run setup in BaseSpace to generate V2 sample sheets. To access the BaseSpace instrument run setup, your BaseSpace account must be toggled to new BaseSpace mode. Once you're on the dashboard, you're going to click on Runs, then New Run, and then select the NextSeq 1000-2000. Next, you will be taken to the Run Setup page. It is important that the analysis location is selected as local when you're using local or hybrid mode. If you select BaseSpace, then the intention is to do the intention was to do analysis locally. Then the file paths, like any genome paths, any bed files, etc., they're going to be incorrect in the sample sheet, and this is going to cause sample sheet validation failure on the NextSeq 2000 control software. So just be very mindful if you're going to do local analysis, make sure you select local for your analysis location. Next, you're also going to want to make sure that you select the correct pipeline that is running on your instrument. So to obtain the version, make sure you go to the DRAGEN settings in the NextSeq 2000 control software to confirm the installed DRAGEN versions that you have. Okay, so after configuring your analysis, you will go to the planned runs page to export the sample sheet. You're going to make sure that the checkbox next to the run is checked off. So here we see that the first run is checked off. Then you're going to click the File button, which is up here at the top. You're going to select Download, and you're going to select Sample Sheet. That will allow your V2 sample sheet to be exported. Then you're going to go ahead and take that sample sheet and you're going to take it to the instrument. So now let's take a look at what a V2 sample sheet looks like. So V2 sample sheets are used only for setting up local runs on the NextSeq 2000. It is correct that hybrid mode also uses a V2 sample sheet, but you will not need to export your V2 sample sheet in hybrid mode because it's going to be connected to your BaseSpace account. So one other note, Illumina Experiment Manager, or IEM, will not be updated to generate V2 sample sheets. So the only way to generate these V2 sample sheets is going to be through the instrument run setup. So now let's just spend a few more moments on the different sections of the V2 sample sheet. So up at the top in teal, we have the read length information. So this is going to describe the number of cycles that are going to be sequenced for your read 1, your read 2, your index 1, and your index 2 if it's dual index. The section in green is going to specify your demultiplexing and FASTQ generation information. So this is going to include your sample IDs, your index or index combinations, and then of course it's also going to include your adapters for your adapter trimming. Now in purple, we have pipeline specific configurations. So for this example, we're kicking off the DRAGEN Enrichment pipeline. So at the top, we have very specific configurations for that pipeline, which includes the reference genome, the bed file, and what type of alignment files we're going to have. Now, next in gray, this section is actually not necessary for local analysis, but I wanted to make you aware that this section does exist. So this is going to be the cloud settings. So if you were to export a hybrid sample sheet, you would see these cloud settings at the bottom. Now, this information is useful, of course, for cloud mode analysis. Okay, so now that we understand what a V2 sample sheet is and how it kind of drives our analysis, let's talk about kicking off a local analysis by requeuing on the instrument. So local analysis can be requeued via the control software process management screen. So to requeue, there's a couple of things we need to make sure. The system state must be idle, so no sequencing or DRAGEN analysis can be in progress. The temporary run folder found in user local Illumina runs must not have been deleted from the instrument. And so analysis cannot be requeued if the temp folder was deleted, even if a user tries to move a copy back into that temp directory. So just be very mindful not to delete that if you need to requeue. And lastly, the sequencing run method must have completed successfully in order to perform a requeue. Okay, so to requeue, you're going to find the run on the process management screen and click requeue. Just note that analysis of runs that errored in sequencing or that were stopped in sequencing cannot be requeued. So in this example, the runs below have an error or stopped status, so the requeue option is faded or grayed out, meaning we will not be able to perform the requeue. When requeuing your analysis, a new V2 sample sheet may be provided and the output directory may also be changed. So to use a new V2 sample sheet, place the sample sheet on a USB or network drive that is accessible to the instrument and browse to it. A new sample sheet may be used to specify a different DRAGEN pipeline than was originally used. So let's just say the first time you did just FASTQ a generation only, but the second time you want to do the DRAGEN-GATK Germline, you will be able to specify that with your new sample sheet. Okay, so let's talk about the output directory for a moment. So if the output directory is not changed from the original location, the original analysis results will not be overwritten. So instead, you're going to get a new subfolder to hold the new output, which will be created in the output directory. So for example, if your original output was your output directory analysis 1 data, then the requeued output directory is going to be output directory analysis 2 plus data. Okay, so now that we understand how to requeue analysis locally on the instrument, let's talk about requeuing a cloud analysis. So the NextSeq 2000 pipelines that were set up in cloud mode can't be requeued. So if your cloud analysis fails or if FASTQ files need to be regenerated in the cloud with a new sample sheet, we're going to use the BaseSpace BCL Convert app. After running the BCL Convert app, you may use the FASTQ files as input for the BaseSpace DRAGEN apps. So a couple of things to keep in mind with these requeue options. First, the BaseSpace BCL Convert app is free to use, but the BaseSpace DRAGEN apps are not free to run. So a BaseSpace subscription is required to run the BaseSpace DRAGEN apps. Now, if a custom genome was used for the initial cloud analysis, the genome will have to be rebuilt using the BSSH Reference Builder app in order to use that custom genome with the BaseSpace DRAGEN apps. And we're going to talk about the two apps later on, and we'll make some distinctions between those apps. Okay, so also to use BCL Convert to requeue, you're going to use a V2 sample sheet generated using the BaseSpace instrument run setup. And now in this case, because it is BCL Convert on the cloud, we are going to need to have those cloud settings or that cloud data section has to be included. So here are some of the BaseSpace DRAGEN apps that can be used once you have the FASTQ files generated by the BCL Convert app. Again, note that to run these apps, a BaseSpace subscription and iCredits will be needed. Also note that when you make a selection for one of these apps, so let's say you select the DRAGEN Germline app, you will have the option to select the different versions that are available on BaseSpace. Okay, so now that we are familiar with requeuing both locally and on the cloud, let's briefly talk about building a custom reference genome to use with DRAGEN. So first, DRAGEN requires that reference genomes are in hash table format. The preloaded genomes that will already be in this format, so we don't have to worry about that. Now, to generate a hash table for a custom genome, the BaseSpace Reference Builder for Illumina Instruments app must be used. This app is free to run. So we just want to make sure that we're selecting that one because there is another app on BaseSpace called the BaseSpace DRAGEN Reference Builder app. Genomes generated with this app are not going to be compatible with the NextSeq 2000 DRAGEN pipeline. So we must use the BaseSpace Reference Builder for Illumina Instruments app if we intend to use a custom genome. So one more time, there's two different apps on BaseSpace to create custom genomes. One of them is going to be specific for the Illumina Instruments, and the other one is going to be the DRAGEN Reference Builder that will be specific for the apps on BaseSpace. So for this particular instance, we're using the one for Illumina Instruments. Okay, so before using the BaseSpace Reference Builder for Illumina Instruments, we need to upload certain files to BaseSpace to build our custom hash table. First, we're going to need the reference genome in FASTA format. This means that the file's extension must be in .fasta or .fa. We're also going to need SAM liftover files. So these files describe the relationship between the primary reference assembly and alt configs and haplotypes. So DRAGEN requires that these liftover files be in SAM or sequence alignment map file format in order for it to be compatible. Now, the last file that we may need is the gene annotation file or the gTF file. This file is optional for all the DNA pipelines, but required for the RNA pipeline. All right, so here's a screenshot of what the reference builder for Illumina Instruments app looks like. Please note the name and the icon highlighted in the yellow box. So the reference builder for instruments, it looks different than the DRAGEN reference builder for the DRAGEN apps on BaseSpace. Another thing to note, the app does say here that there is a computing cost associated, but the app is actually free to use for any account here. So whether you're basic, professional, or enterprise, you would be able to launch this app if you need to create a custom genome. Once you run the app in the instrument run setup, both the standard or the built-in genomes and your new custom genome will be available to select from the dropdown. Okay, so now that we understand how to build a custom genome, let's talk about using that custom genome for local analysis. So in this situation, you built your genome on BaseSpace using that free app, and now you're going to take that genome and you're going to put it into the instrument for analysis, for local DRAGEN analysis. Okay, so first, the output of that reference builder for Illumina Instruments app is going to be in tar.gz format. This file must be downloaded from BaseSpace. So make sure to save that tar.gz file to either of the following locations. Now, these two locations are also listed in the system guide, so if you don't remember, we can always check there. Now, the first location that you can save that tar.gz file, is the temp folder on the NextSeq 2000. The second location is to a mounted network drive that has accessibility to the instrument. So storing the file in a different location, so not the temp folder or a mounted drive with access, can result in unable to access file errors when you're trying to import the genome. Now, for cloud analysis, I'm just going to say this here. For cloud analysis, that tar.gz file does not need to be downloaded at all. It's going to exist in the cloud, and it doesn't have to make a stop at the control software. Okay, so once you have the tar.gz file, let's talk about importing it through the control software. First, you're going to log into the NextSeq 2000 as Illumina admin. In the NextSeq 2000 control software console, you're going to go to the DRAGEN page. Then next to genomes, you're going to click the import button, and you're going to go ahead and browse to that tar.gz file that you downloaded from BaseSpace. Once the import is successful, you're going to see this page informing you that you were able to successfully load that genome. Okay, so here we're just showing a list of all of the preloaded genomes that already exist on the DRAGEN server. So the hash tables are already built for these. We wouldn't have to go through building any of these out. But if you do have a particular genome of interest, again, you're going to follow those steps so that you can perform your analysis with that genome. Okay, so at this point, we've basically covered all of the run setup scenarios. So in the next portion of the webinar, we're going to discuss finding the output files, the DRAGEN pipelines themselves, and we're also going to talk about, we're going to end the webinar with where Universal Copy Service transfers all of the DRAGEN output files from the instrument. Okay, so let's talk about the output files and where we're going to find them when we set it up for cloud analysis, local analysis, and local analysis to upload to the cloud. All right, so when analysis is performed in the cloud, the DRAGEN output files are found within the analysis app session. So a link to the analysis session can be found in the run summary page, and here we're seeing it on the lower left corner. The analysis session can also be accessed from the project associated with the run. So for cloud analysis, the output files include everything, the FASTQ and BAM files for all the DRAGEN pipelines. Now, when analysis is performed locally and saved to a local output directory, for example, your network drive or an external drive, DRAGEN output files are going to be found in the analysis folder within the run folder. In local analysis, FASTQ files are saved and transferred to the local output directory only when the DRAGEN FASTQ generation pipeline is ran. After local analysis completes for pipelines other than the DRAGEN FASTQ generation app, the FASTQ files will not be found in the temp run folder of the instrument, nor will they be found in the output folder. So if you need FASTQ files to be saved and transferred to your local directory, you're going to have to run the DRAGEN FASTQ generation pipeline via a requeue, like what we described before. Okay, when analysis is performed locally and then uploaded to the cloud, the DRAGEN output files are uploaded to a project named for the run. So clicking the link to the upload session or the app session, again shown here on the lower left, will take you to the app session within the project. So within the app session, within the project, we're going to click on the Files tab to find our output. The output includes variant calling and RNA-quant and RNA fusion output, depending on the pipeline that was ran, and of course the log files and metrics. When analysis is performed locally and then uploaded to the cloud, the following DRAGEN output are not uploaded to the cloud. So FASTQ files are not uploaded to the cloud for any pipeline, including the DRAGEN FASTQ generation pipeline, and neither are BAM files. So if you need FASTQ files and or BAM files to be stored in the cloud, then NextSeq 2000 must be run in cloud mode instead of in local mode uploading to the cloud. So the alternative, if the run was uploaded to the cloud for run monitoring and storage, the FASTQ files can be generated in the cloud using the BCL Convert app. Okay, so now that we have an understanding of where to find our output files and what files are going to be present, let's talk in detail about the actual DRAGEN pipelines. So first, let's start with the DRAGEN FASTQ generation pipeline. So DRAGEN FASTQ generation uses BCL Convert to demultiplex, adapter trim, UMI trim, and generate our FASTQ files. The input is going to be the run folder and, again, that V2 sample sheet. And of course, your output are going to be your demultiplex FASTQ files and all of the stats files associated. So let's touch upon the configurable settings. So first here in the blue box, we have the adapter trimming settings. So if you selected an Illumina index kit, you will not need to configure this setting. It will be on by default. However, if you did use a custom index kit, here's where you would indicate the adapter sequences to trim off of the 3' end of the reads. So the only characters that are allowed here are capital A, capital T, capital C, and capital G. Next, in the green box, we have our barcode mismatches setting to configure. So by default, the setting is set to 1, meaning to allow 1 barcode mismatch. If you would like to be more stringent, you can set it to 0, meaning that the index sequences must match exactly for a read to be assigned to the sample. Or if you want to be less stringent, you can allow up to barcode mismatches of 2. So again, what that means is that you can allow up to 2 mismatches in the index or index combination and still get a read assigned to your sample. Now, lastly, we also have the override cycles option in the purple box. And this option is mainly for our UMI sequence and UMI settings. So I'm going to give you a full example of that in the next slide. So to illustrate this example, let's consider an example run. So this example run is going to be a 2x151, and it's going to be dual index 8 base pairs. So in this scenario, the instrument collects and stores 318 cycles worth of data, which is going to generate cycle folders C1.1 to C318.1. Now, by default, as is written into the runinfo.xml file, BCL Convert is going to treat the cycles in the following way. So cycles 1 through 151 are going to be considered read 1. Cycles 152 to 159 are going to be index 1. Cycles 160 to 167 are going to be index 2. And cycles 168 through 318 are going to be read 2. So now the override cycles value is going to look like this. So Y151, I8, I8, Y151. Now, let's say that the first 7 bases of read 1 and read 2 are UMIs, and the 8th base is going to be a linker that we're going to want to ignore. The override cycles has the following symbols that are going to allow us to perform just that. So the first symbol is going to be Y, and that represents data, which means we're going to write it to the FASTQ file. N means ignore, and we're going to exclude anything with an N from the FASTQ file. I is going to represent an index read, and that's going to be used for demultiplexing. So if we're identifying a cycle as an index cycle, that has to be accounted for in the sample sheet. And now U is going to stand for UMI. And what this does is when we indicate that a cycle is going to be a UMI cycle, it's going to take that sequence and add it to the header of the FASTQ file. So for this specific example, our override cycles value is going to look like this. U7 for the first 7 bases of the UMI in read 1, N1 to ignore that 8th base or that linker sequence, and then Y143, which is going to be our actual read 1 data. Next, we have I8, I8, so that's going to be our dual index, so the sequence that's going to be used for demultiplexing. And then again, for read 2, we're going to have that U7, again, to represent the 7 bases that are going to be UMI, N1 to ignore that 8th base or that linker sequence, and Y143, which is going to be our read 2 data. So this override cycle setting is very similar to the use basis mask setting, which was available in BCL to FASTQ. Okay, so now let's talk about some of the stats files that get generated with the FASTQ generation pipeline. The first one is going to be the index metrics out.bin, which is a binary file that contains a summary of the demultiplexing results. So the contents of the index metrics out.bin are visualized either in BaseSpace in the Run Indexing QC tab, and that's what we have an example of here, or in the SAV Indexing tab. Now, for cloud, hybrid, and local runs that use cloud storage, the Indexing QC chart in the BaseSpace Run Summary tab is going to populate once the run completes and all of the files have been uploaded. For local runs that use only local storage, the Indexing QC metrics can be viewed in SAV, and we're going to take a look at that in the next slide. So here we have a sequence analysis viewer, and we're showing the indexing tab that gets populated from the index metrics out.bin. So for local runs that use local storage, the indexing QC metrics can be viewed in SAV. And to view them in SAV, we have to do the following, steps just to make sure that we can see them. So we're going to copy the index metrics out.bin file from the report folder to the level of the interop folder. So the reports folder path is going to be inside of the run folder, analysis, data, the number, reports, and we're going to copy that index metrics out bin file and move it into the interop folder, which is going to be inside of the run folder. Then you're going to launch SAV and browse to that run, and you'll be able to populate that tab. Okay, so some other useful stats files that get produced with this as well are going to be the demultiplexing stats.csv and the top unknown barcodes.csv. So now the first one, the demultiplexing stats over on the left, it's going to list the number of reads that got assigned to each of our index combinations that we included in our sample sheet. We're also going to get the number of reads that were assigned to the undetermined. Now, if you encountered some type of demultiplexing issue where your undetermined is much bigger and your expected indexes did not get as many reads as expected, you can then use the top unknown barcodes.csv file. So this file is going to essentially give you exactly what the instrument read, and it's going to give you a readout of the number of reads that were assigned to it. So if you are in a position where you have some type of demultiplexing issue, you can definitely check the top unknown barcodes and you can see whether maybe you have your index 2 in the reverse complement orientation, or maybe you just have the incorrect indexes or whatever the case may be. You can definitely find that using the top unknown barcodes file. Okay, and lastly for this pipeline, I wanted to talk about some of the hard-coded options or non-configurable options. So the very first one, this use strict mode is going to be set to false. And basically what that means is if the software encounters a corrupt or missing BCL file, it's not going to terminate the analysis, it's going to continue on, and it's going to still produce your FASTQ files. Now the adapter behavior is always set to trim, which means that we're always going to be doing adapter trimming when we generate our FASTQ files. Two other settings that we talk about are going to be the minimum trimmed read length of 35 and the mass short adapter reads of 22. So these kind of work together, so I'm going to talk about them kind of together. So the first part is if you adapter trim and you end up with a read that is below 22 base pairs, that mass short adapter reads is going to tell the software to add ends until we make it the minimum trimmed read length, which is set to 35. So again, if you adapter trim, your read is less than 22, the software is going to add ends until we reach 35, so you'll end up with a read of ends. Again, that's only for a scenario where your read is very, very short. All right, so we covered the FASTQ generation pipeline, so now let's jump over and talk about the DRAGEN-GATK Germline pipeline. So this pipeline, the main goal is to perform alignment and variant calling, and this is whole genome alignment and variant calling. So your FASTQ files are going to be used as input. The DRAGEN aligner is then going to align them to the reference genome, and you're going to get your BAM or CRAM files. Once you have your BAM and CRAM files, whatever you decide, we're going to run those through various variant callers. So let's talk about those. So the first one is the DRAGEN Germline Small Variant Caller, or the SNV and Small Indels, for SNV and Small Indel detection. So again, we're going to note that even though the pipeline is called the DRAGEN-GATK Germline pipeline, the GATK Small Variant Caller is not used. Also, we need to note that the DRAGEN Germline Variant Caller assumes a diploid genome. The next variant caller that we have is DRAGEN Manta, which is going to be our structural variant caller. So this is going to be able to detect deletions, insertions, tandem duplicates, intra and interchromosomal translocations, and complex structural variants. The next caller is going to be the DRAGEN CNV caller, or the copy number variant. So it's going to detect copy number variations. Next, we have DRAGEN Expansion Hunter for repeat expansion detection. So this is going to detect expansions of short tandem repeats, or STRs. These repeat expansions are responsible for diseases like Fragile X syndrome and Huntington's disease. And the last caller that we have available is going to be the ROH caller, or ROH region identification. So runs of homozygosity on autosomes or contiguous lengths of homozygous genotypes. So this is present in an individual whose parents were transmitting identical haplotypes to their offspring. So ROH applications are going to include studying things like disease risk. Okay, so like the previous pipeline, we also have some configurable settings for the DRAGEN-GATK Germline pipeline. The first option is going to be your reference genome. So you can select from any of the pre-built reference genomes, or if you did create a custom genome, you will have the option to select it here. And then the next portion allows us to decide whether we're going to generate BAM or CRAM files. One thing to note, if you selected local analysis, so in the previous page, you have the option to select between local and BaseSpace, and if you picked local, you'll also have the option to not generate any BAM files. So here's just a list of the pre-built genomes that are going to be available for selection under the Germline analysis. And next, we're just going to talk about the hard-coded Illumina DRAGEN-GATK options. So really, the main hard-coded options that I wanted to mention were that duplicate marking, BAM indexing, BAM sorting, and assay tags for recording chimeric and assay alignments. Those are all going to be set to true. So these are going to be considered our standard settings. Now, another thing that I want to note is that the option to annotate variants using the Illumina annotation engine, or Nirvana, is not enabled. So variants in the VCF and gVCF files will not be annotated. So some other files that get produced on top of your BAMs and VCF, you're going to have your mapping metrics.csv. So this is going to give you your alignment metrics, such as your percent aligned or your mapped reads and your percent duplicates. You're also going to have your coverage metrics.csv, which is going to give you information such as average coverage over the genome and the percent of the genome that was covered at least 20x or 50x, etc. Okay, so let's briefly talk about the DRAGEN enrichment pipeline. So as we had mentioned earlier, this pipeline is very similar to the GATK Germline pipeline in the sense that it's going to perform alignment and variant calling. However, our variant callers are now going to be limited to the DRAGEN Germline variant caller for your small SNVs and indels and the MANTA structural variant caller. One other thing to note here is that we are going to need a bed file, and what this bed file is going to allow for is for the variant calling to be concentrated in the regions indicated. So let's just talk about the bed file for a moment. So a bed file or a browser extensible data file is going to contain a list of the targets for the enrichment library prep. As we see here in purple, we have the chromosome. In the blue, we have the target region start and stop coordinates. And in the green, we have the target region name. So basically what's going to happen is after alignment occurs, the bed file is going to be used during variant calling so that we can hone in our variant calling into these exact regions of the, we're going to want to do variant calling within the target regions listed here in this bed file. Another thing, our alignment and enrichment metrics are all going to be calculated based on the alignment to these target regions in the bed file. So again, we have configurable settings that are very similar to the DRAGEN-GATK Germline pipeline. So you're going to have the option to select your reference genome. You can select whether you want BAM or CRAMs, or if you want none when selecting local analysis. And then for your target regions, any Illumina enrichment kit, the bed file is going to be available here on this dropdown. Now, if you used a custom kit or something else, you can select the custom bed file option here, and it will allow you to upload it. And it will work as long as it has the right format. So again, just a list of all the genomes available for analysis with this pipeline. And one more time, we have the hard-coded DRAGEN enrichment options. Now, these are going to be exactly the same as the Germline GATK pipeline. So again, we're going to enable duplicate marking, BAM indexing, sorting, and we're going to enable those assay tags for chimeric and assay alignments. And just like with the previous pipeline, variants are not going to be annotated with Nirvana. So in the VCF and gVCF files are not going to contain any annotation information. We also have important metrics files. So again, we talked about the QC coverage region cov report.bed. So this is going to list our mean coverage per target region. So it's going to give you the statistics about what was covered in your target bed file. Now, the summary.csv file, also very useful. It's going to include your alignment metrics, your variant calling metrics, and it's going to include coverage metrics. Now, one important metric for enrichment analysis is going to be uniformity of coverage, which is the percentage of target regions covered at greater than 0.2x the mean coverage. Okay, so let's just briefly talk about our last pipeline, which is the RNASeq pipeline. So the DRAGEN RNA pipeline quantifies gene expression. So it determines the relative abundance of different RNAs. The DRAGEN quant module counts or quantifies reads aligned to the genes and transcripts in the reference annotation and normalizes those counts using the TPM or transcripts per million mapped reads method. The DRAGEN RNA pipeline does not perform differential expression analysis. So you can use a third-party tool such as DESeq2 for DESeq analysis. Now, if you're using BaseSpace or if you're using cloud mode, you can use the DRAGEN differential expression app, and that will give you your differential expression results. Now, one other thing to note, the gene annotation file for this pipeline must be in GTF or GFF format. GFF3 format is not going to be supported. So for fusion detection, gene fusions are called based on reads mapping to or split between two different genes. These split reads are chimeric reads or supplementary alignments that are going to have that SA tag in the BAM and CRAM file. The fusion detection module is not looking for predefined fusions, so it is possible to detect novel fusions with the fusion detection. So again, we have our DRAGEN RNASeq pipeline configurable settings. This is going to include selecting your reference genome, deciding between BAM and CRAM. And now we are going to have this third option, which is going to be the annotation file, which I'm going to talk a little bit more about in the next slide. So the annotation files provide our exon boundaries to the aligner when gene locations for quantification and gene fusion detection. So when uploading a custom annotation file, the file must be in GTF. So GTF is the gene transfer format. So if you're using a preloaded genome like the human, mouse, or rat genome, and a custom file is not uploaded, the annotation file within those predefined genomes is going to be used instead. Now, if you want to find your own annotation files, you can certainly get them. There's resources like RefGene, RefSeqGene, Ensemble, UCSC, GenCode, AceView. These are all places where you can get these types of files for analysis. And for this pipeline, again, we just have a list of the genomes that are available. And again, we have our DRAGEN hard-coded non-configurable settings. So the settings here are going to be very similar to the previous two pipelines in that BAM, indexing, sorting, and the SA tags are going to be set to true. But one thing to note is that duplicate marking is set to false, and this is typical for RNA-seq type data. Okay, so the main files that we're going to get, we're going to get our quantification results, which are going to be expressed in or normalized in TPM. So transcripts per million mapped reads, it's a normalization method that's very similar to FPKM. So you're going to get both your quant.sf, which is going to be your transcript level quantification results, and your quant.genes.sf, or the gene level quantification results. You're also going to get your fusion candidates final file, and you're also going to get your mapping metrics.csv. And again, just a reminder, those mapping metrics are not going to include duplicate marking because that setting is not configured, but it will contain your alignment metrics like your percent aligned reads and total chimeric alignments. Okay, so now that we went through all of the pipelines, let's just briefly again revisit what files you're going to get in your local and hybrid analysis transferred by UCS. We've been talking about this quite a bit throughout the presentation, but here we're just going to formally describe everything. So for local or hybrid modes, the DRAGEN FASTQ generation pipeline generates our FASTQ files on the instrument. So UCS transfers the FASTQ files to your local storage if your local storage is specified. Now, UCS does not transfer the FASTQ files to the cloud, even when the UCS cloud for run monitoring and storage is selected. So again, if you really need your FASTQ files and you need them in the cloud, we recommend running it in cloud mode rather than in local mode, sending it to the cloud. Now for the DRAGEN-GATK Germline pipeline, the FASTQ files are not going to be transferred by UCS, neither to the cloud nor to your local storage. So if you really need your FASTQ files in the cloud, again, we're going to recommend that you set this up in cloud mode. If you really need it in local storage, then you're going to requeue the run using the DRAGEN FASTQ generation V2 sample sheet. We have the same thing going on for the DRAGEN enrichment pipeline. So if you're sending a run, if a run was analyzed locally and you're going to send that to the cloud, the BAM and the FASTQ files are not going to get transferred. So again, we want to make sure we set that up in cloud mode if those are files that you're looking to have in the cloud. Now for your local storage, the BAM files will be transferred, so your alignment will be in the final storage location, but your FASTQ files will not be. So like with the GATK Germline pipeline, you're going to have to requeue that if you need the FASTQ files in the local storage. And we have the same exact scenario for the RNASeq pipeline. So if the RNASeq pipeline is done locally and you're going to send this data to the cloud, you're not going to have your alignment or your FASTQ files. Of course, you'll have all of your other files that you may need, but if you need to perform any type of troubleshooting or if you want those files, we really recommend that you set this up in cloud mode so you have that available. And similar for the local storage locations, the FASTQ files are not going to get transferred, so if you need them, you'll have to generate them using our FASTQ files. I'm sorry, using our FASTQ generation pipeline. Okay, so that brings us to the end of this presentation. I want to make sure that we get to some of the questions.